Have you ever wondered what the secret blueprint of life looks like? What if I told you that one of the biggest scientific breakthroughs of the 20th century was made by a young, ambitious American who was just in the right place at the right time? This is the story of James Watson and the race to uncover the structure of DNA. James Watson was in Chicago in 1928. From a young age, he was a true prodigy, a brilliant birdwatching enthusiast who enrolled at the University of Chicago at the tender age of 15. He was fascinated by genetics and the fundamental questions of life. After earning his PhD, his quest for knowledge led him across the Atlantic to Europe, first to Copenhagen, and then, fatefully, to the Cavendish Laboratory at Cambridge University in England in 1951. It was here that his life would change forever. At Cavendish, he met Francis Crick, a sharp-witted physicist who was 12 years his senior. Despite their different backgrounds, Watson, the young biologist, Crick, the seasoned physicist, they instantly clicked. They shared an intense curiosity and a bold conviction that the structure of the DNA molecule was the most important scientific puzzle of their time, and they were the ones to solve it. At the time, scientists knew that DNA carried genetic information, but no one knew how. What did it look like? How did it copy itself? Watson and Crick decided to take a different approach from their competitors. Instead of spending endless hours on difficult experiments, they focused on building physical models. They gathered every piece of data they could find, like clever detectives putting together clues from different sources. A crucial piece of the puzzle came from the work of Rosalind Franklin and Maurice Wilkins at King's College London. Franklin, a brilliant X-ray crystallographer, had produced an incredibly clear image of a DNA molecule, famously known as Photo 51. When Watson saw this image, it was a eureka moment. The pattern strongly suggested a helix, a spiral staircase shape. Another vital clue came from biochemist Erwin Chargaff, who had discovered that in any DNA sample, the amount of adenine A always equaled the amount of thymine T, and the amount of guanine G always equaled cytosine C. This became known as Chargaff's rules. Putting it all together was like solving the ultimate 3D puzzle. Watson and Crick realized the structure had to be a double helix, with two strands twisting around each other, using their knowledge of chemistry. They figured out that the A on one strand always paired with the T on the other, and the G always paired with the C. This elegant pairing explained Chargaff's rules perfectly and also suggested a brilliant mechanism for how DNA could unzip and copy itself. In 1953, they announced their groundbreaking discovery in a short, one-page paper in the journal Nature. It was a monumental achievement that opened the door to modern biology and genetics. Their double helix model has since become one of the most iconic symbols in all of science. In 1962, Watson, Crick, and Wilkins were awarded the Nobel Prize for their discovery, a testament to the power of collaboration, intuition, and a relentless desire to understand the very essence of life. Thank you so much for watching. If you found this story as fascinating as I do, please hit that like button and subscribe for more deep dives into the history of science. See you in the next video.